Welcome, everybody. It's wonderful to see you all in person. It's really a delight, and welcome to Transforming Healthcare. This program highlights new directions and innovations in healthcare, in which the program also immodestly showcases the remarkable achievements of our faculty. So it's a real pleasure for me to welcome you to this evening's events. Tonight we're going to be talking about RNA. I'm Peter Buttrick. I'm the Senior Associate Dean for Academic Affairs, and I'm apparently your MC for the evening, and I'm actually privileged to have that distinction. Kicking off tonight's program is Don Elliman, who's the Chancellor of the University of Colorado Anschutz Medical Campus. This public lecture series began about 6 years ago with support from the Chancellor's office. So appropriately, Don gets credit for the vision behind the program. And of course, his office continues to provide support, for which we are very, very grateful. Please join me in welcoming Chancellor Elliman.
Thank you, Peter, and thank you all for coming. I mean, this is the first time many of us have seen each other in 2 years, and that makes it pretty special in and of itself. But I think you're going to find tonight's subject fascinating. As Peter said, the title of tonight is The Proof and Promise of RNA Research. And obviously many of us have heard, in most cases, I think, out of the blue about RNA over the last 2 years. And I think we're going to find it fascinating to hear more about it and more about the breadth of it than we have strictly as it related to COVID. I'm also welcoming you to our new building, to our kind of our front door, if you will. We started this building almost 3 years ago. We moved our first faculty and staff in about 2 months ago, and it's for us, I think, a recognition that we're turning a corner. The campus has been on a growth curve that's exponential. We desperately needed new space, and we decided we'd break away from the red brick mold and design something that was a little different and hopefully one that speaks to the future of the Anschutz campus, which is a very bright future. And you're going to hear about why that's a bright future tonight. You know, we often talk, every other session we've had in this series has been on clinical activity or clinical science. We have not. This will be the first one we've hosted on basic science. And I think it's going to be fascinating for all of us to hear about how the basic science foundation that we build here on this campus is what eventually gets translated into the bedside. And that's really what tonight is about in many, many ways. So enjoy the panel. Thank you for your support. We couldn't have done what we're doing today without supporters like you and without people who are interested in what we do like you are. So welcome, enjoy the evening, and I'll turn it back to Peter to introduce the panel.
Actually, we're turning the program briefly over to Dean John Riley, who's on an airplane flying someplace at the moment, but wanted to provide some opening remarks.
Hi, I'm John Riley, Dean of the School of Medicine at the University of Colorado, and it's my pleasure to welcome you to this program related to the RNA Biosciences Initiative. When I arrived here as Dean in 2015, one of the early programs we undertook on this campus was an internal research competition to fund exciting projects to move fields forward. One of the winners of that competition was the RNA Biosciences Initiative. It was chosen because we already had an outstanding nucleus of investigators here who were doing cutting-edge work in the field. There was a long track record of productive RNA research in the University of Colorado system here in Colorado, and they outlined ambitious plans to recruit faculty, train graduate students and MD, PhD students, and to put on a series of seminars to create a community around the field of RNA bioscience. Now, the events of the last 2 years have catapulted RNA into the public lexicon with the rollout of RNA-based vaccines for the COVID pandemic. And that technology was built on years of research that took place over the past several decades. Our goal is to fuel research that will provide the kind of platform for translational research and therapeutics in the upcoming decades. The track record of this initiative over the last 5 years has been very, very impressive. They have recruited outstanding young faculty, some of whom you will hear from today, have established a critical mass and outstanding community, and fostered cutting-edge research. We had the pleasure of having a distinguished senior member in the field of RNA bioscience here as our distinguished dean's guest lecturer several months ago. And she remarked, compared to a decade ago on her last visit to this campus, that the RNA community here has been absolutely transformed. I hope you will take away from this session an appreciation for the outstanding work that this group of investigators is doing. One of the unique features of the University of Colorado School of Medicine is a faculty culture that supports and welcomes the investment in the basic science community. I hope you recognize the talents of the individuals you're going to hear today, appreciate the cutting-edge work that they're doing, and join me in thanking them for advancing the field forward. And again, I want to thank you for attending this program.
Thanks, Chancellor Elliman. Thanks in absentia, Dean Riley. I don't want to minimize those opening remarks, but now we're going to get to the substantive portion of the program. RNA biology broadly is one of the single most exciting areas of investigation in the scientific world today. And it's a wonderful example of how basic investigation has led to transformational clinical advances. And as I think you'll hear this evening, that remarkable trajectory is certain to continue. Our 5 speakers are all members of the RNA Biosciences Initiative, and all are exceptional scientists and innovators. Parenthetically, it's always fun for me to introduce members of our research community because they're always very enthusiastic and excited about the work that they do. But the RNA people are on the extreme end of that enthusiasm spectrum, so you're going to have a fun evening. Our first speaker is David Bentley, who is the co-founder and current co-director of the RNA Bioscience Initiative. David is a professor in the Department of Biochemistry and Molecular Genetics. As a preface, let me just remind you we're going to have a question and answer period at the end of the 5 presentations, so hold your questions till the end. And now, welcome Dr. Bentley.
Thank you, Peter. Thank you, Chancellor Elliman. And thanks to all of you for coming out this evening to this beautiful new building. So we at the RNA Biosciences Initiative are very excited, a bit nervous, and absolutely delighted to be able to introduce to you this evening one of the brightest stars in life's galaxy, the very talented, the very beautiful molecule of RNA. So we at RBI are very lucky people. We get to spend our time studying the biology of this marvelous molecule, whose abilities seem to never cease to astound us, and we get paid to do it. So first and foremost, research scientists are curious people. They are always asking questions. Questions like, how does this work? Why does it work this way? What does it look like? Can't we look at it in a little bit more detail? It can get a little bit annoying even. We, like people in other professions, make things. But rather than making, hamburgers or financial deals or litigation or legislation, we are in the business of making discoveries. Discoveries about how life on this planet works. And the way we do that involves this beautiful process of basic research. Basic research allows someone driven by their curiosity, empowered by their ingenuity and their creativity, using their intuition, their head, their heart, their hands, to persuade Mother Nature to reveal secrets about how life works. And there's nothing more exciting in the life of a research scientist than the instant when they realize they're the first person in history to have understood this secret that Mother Nature has revealed to them about the puzzle of how this wonderful thing called life works. So epiphanies like that don't happen every day in the life of a scientist. You're fortunate if it happens on a few occasions in your entire lifetime. But when it does, I would say it's a spiritual experience, and it's an absolute thrill. So I know that the stereotype of the research scientist might be something of an egghead, and I'm not saying that's totally, doesn't have some element of truth to it. But I want you to remember that on the flip side of that coin is a thrill seeker. So to give you a little bit of insight into this enigmatic character, part egghead, part thrill seeker, I want to tell you about my hero in science. He's a man named Fred Sanger. He was the director of the department where I did my PhD studies in Cambridge in the UK a long time ago. One of my few close encounters with Sanger was in the fall of 1980. I was in the second year of my PhD work. I'd come into the lab early on a Sunday morning. That was the era in my life when, I was never at my best on a Sunday morning. They tended to come after a Saturday night, It was maybe a little bit the worse for wear. I get into the elevator, and Sanger steps in there with me. It's a small gray steel box that moves very slowly. I'm in there with this guy who I'm, a little bit afraid of. I'm kind of in awe of. I'm sure he doesn't really know who I am. We go up to the top floor at a glacial pace. I'm kind of leaning against the side of the elevator, staring at my shoes. And at the point when the silence becomes, truly painful, Sanger broke the silence by kind of mumbling to me, "Congratulations on your prize." So my first reaction was shock because I had no idea he knew who I was. The context here is that the previous week I had been awarded a prize of 60 pounds to travel to a scientific meeting. The week before that, Sanger had also been awarded a prize. He'd been awarded his second Nobel Prize. So you can see him here. You can see also that security at this event clearly wasn't very good because this character somehow got in there who's clearly only there for the free champagne. Sanger won his second Nobel Prize for discovering how to extract the information stored in the genetic material, DNA. He developed the technology that made the Human Genome Project and personalized medicine possible. So he's my hero. He's an inspiration to me. His portrait is on the wall in my office, not really because he made 2 trips to Stockholm, but because he personifies the awesome power of basic research. Here was a man who was humble to a fault, not an impressive public speaker, certainly not someone who wore his genius on his sleeve, but who, through the power of basic research, was able to cozy up with Mother Nature, and have her essentially hand over the keys to the crown jewels, allowing us to unlock the information in our genomes. A discovery that has changed the world, has impacted the lives of thousands of people in ways that Sanger couldn't possibly have imagined at the time he made the discovery, and in ways that are going to transcend his lifetime by decades. This is the power of basic research, driven by a man's curiosity and ingenuity. So I'm feeling now as if I've, was the host at the Oscars who spent way too much time introducing the star who's getting the award. So without further ado, I want to show you, to introduce tonight's main attraction, RNA, with a short video that we've made. My name's David Bentley. I'm a researcher at CU Anschutz, and today I'm going to tell you about RNA. Inside every living organism, you'll find both DNA and RNA molecules. They're both essential for all life on Earth, and every cell has thousands and thousands of RNA molecules. You probably know about DNA, the double helix. It's a very stable, linear molecule and serves as a safe repository for the genetic information that we inherited from our parents. By contrast, RNA is a short-lived molecule that's constantly being made and destroyed. RNA molecules carry genetic information. It's kind of like those letter banners that you string together and hang up at a party that read something like "Happy Birthday." RNA is a string of letters, but instead of 26 letters, RNA has only 4, G, U, A, and C. And the order of those letters has meaning. RNA not only carries information, it also puts that information to work by directing operations in our cells. Each RNA molecule uses its unique combination of letters to make our cells do all sorts of useful and important things, like produce proteins or catalyze chemical reactions. Now here's where the COVID-19 vaccines come in. They're made of a type of RNA called messenger RNA, or mRNA. That's because it carries a message with precise instructions on how to build a harmless protein that's unique to the COVID-19 virus. The shot in your arm delivers the mRNA into your cells, where it starts directing the production of the protein. The body then responds by creating antibodies against the protein. Those antibodies are like warheads that lead the attack against the virus, so our immune systems can shut down the infection. The COVID-19 vaccine is the culmination of decades of RNA research, starting back in the early 1960s, and some of the most important discoveries in RNA research were made right here at the University of Colorado. Today, we're studying new ways to use RNA to treat many other diseases, so now is a particularly exciting time for our research team. The development of the messenger RNA-based vaccines against the SARS-CoV-2 virus that causes COVID-19 are spectacular achievements of translational researchers. Translational research is highly focused on development of therapies and cures for specific diseases. The relationship between translational research and curiosity-based basic research, I think, is kind of like the relationship between me and my sister when we were kids. You see, my sister was a ballerina. I was the kid who got A on the science test. So you can kind of imagine who got all the parental attention. But it's totally okay. I'm over it. I am. But like a ballerina, translational research requires a partner to ground her, to lay a foundation. And that is the role of basic research, often operating not in the spotlight, but serving an essential purpose role in enabling important advances in medicine. Many of these advances in basic research in RNA have been made here in Colorado. Tom Check, working in Boulder, Sidney Altman, who actually spent a couple of years as a young scientist here at the med school, rocked the foundation of biochemistry in the 1980s by showing that RNA could catalyze chemical reactions, a staggering achievement for which they received the Nobel Prize in 1989. Cutting-edge basic research in RNA continues today at the RNA Biosciences Initiative, and I'm very lucky to work with a dream team of researchers in this area there. Before I leave you, though, and you're going to meet some of those, some of my colleagues in a few minutes. Before I leave you, I know that there are movers, shakers, influencers in the audience, and I want to plant with you the seed of an idea. I believe that it's time the state of Colorado claimed her rightful state molecule. And that molecule should be damn right. So now I'd like to hand over to my friend, Jeff Kieft. Jeff is a world authority in the field of RNA structural biology, which you're going to learn all about. He came to us here via West Point, Yale, and the White House Office of Science. Jeff.
Well, thank you very much, David, and thank you, all of you, for being here this evening. It's really wonderful to see so many people excited to learn about RNA, and hopefully we can deliver the goods and you'll leave even more excited than you arrived. So let me start out by pointing out something that's probably becoming apparent to many of you already, and that is that RNA is a remarkably versatile molecule. It's essential for life as we know it. It can be the causative agent of disease, and, as you're going to hear more about tonight, it can be the basis for new therapies. So that's extremely exciting. But it also, to me, presents a little bit of a paradox. Because what you've also just heard is that RNA, or the alphabet of RNA, consists of only 4 letters, G, C, A, and U. So how can a molecule that appears on its surface so deceptively simple be capable of all of the functions that you're just starting to hear about tonight? A hint to resolving this paradox actually can be found in the world that you encounter every day. And in that world, the objects that you work with, that you touch, that you use on a daily basis have a given function. They work the way they work because of their structure. So let me give you an example. So many of you probably have already done spring planting, right? Put in some flowers in the yard. When you did that, you grabbed a shovel, something that looked like that. Why did you use a shovel? Because it has the correct 3-dimensional structure that if you hold it in your hand, you can dig in the dirt. You didn't grab a hammer, you didn't grab a saw, because although they are made of essentially the same materials, they don't have a 3-dimensional structure that is conducive to digging. The same thing holds true in the biological world. The bones in your arm and your hand have a different structure, a different configuration, than the bones in a bird's wing. They're both made of bones, but the different structures give rise to different functions. And so the implications of this are very, very important. What it means is if you can study the structure of an object and really analyze it, it can tell you a tremendous amount about what it does and how it works, and maybe how you could even improve it or adapt it or further engineer it, right? Engineers do this all the time. They may look at the schematics of something like a rocket engine or take that engine apart and reverse engineer it and figure out how it works. And paleontologists might do this by looking at the skulls of dinosaurs to figure out, well, the structure of these teeth means this one was a meat eater. The structure of these teeth means that one was a plant eater, right? So the structure-function relationship is all around us, and we can extend it down to the level of molecules. So in the case of RNA, you've already heard that we can think of RNA as a string of letters, but it really isn't a string of letters. That's a representation of the molecular structure. If we could look at what this RNA molecule really looked like, look at the molecule itself, you would see something that looks like this. This is the three-dimensional structure of the RNA that is depicted by this in this sequence at the top of the slide. And it's the three-dimensional structure of this RNA that gives it its function. Different sequences of RNAs will have different structures and therefore different functions. So you probably haven't thought of RNA as this twisted up, contorted, complex shaped molecule, but that's what they look like. And if we can study them at a detailed level, that can give us powerful insight into how they work and then how we might use them in the development of new therapeutics. The problem is, nobody can just look at an RNA sequence like this and reliably tell you what the structure is going to look like or what it's going to do. We need to do that experimentally, and that's extremely difficult. What we really need to do is see the three-dimensional structures of many, many, many more RNA sequences. I would say thousands more RNA sequences to understand the basic rules by which this gives rise to that structure that I just showed you and creates all the versatile functions that RNA is capable of. And so what I want to talk about for the rest of my presentation is really two things. One, where can we find interesting, important RNA structures to study? And two, how do we go about actually looking at molecules? So the first question, where can we find informative, biologically important RNAs to study? To answer that, I'm going to blow your minds a little bit, okay? You ready for some really big numbers? This is an astronomically big number. That's a lot of zeros, right? And in fact, that is the number of stars estimated to exist in the observable universe. All of the galaxies, all of the pictures taken with the Hubble Space Telescope, this is how many stars they think are out there. This is beyond astronomically large. It is much, much, much larger than this number. That is the number of infectious viral particles that currently exist on Earth. Everybody reach for your hand sanitizer. No. I'll make you feel a little better about this number by pointing out that the vast majority of these belong to viral species that do not infect humans. Now, you may not have thought about this, but every form of life on Earth is infected by viruses. Bacteria have viruses that infect them. Sea slugs, oak trees, elephants, ants, they all are infected by viruses. And so these viruses surround us, they're all over the Earth, and they're constantly, I guess you could say, doing battle with the organisms that they infect. And so, I'll also point out that most of them are not of any danger to us, but we can learn from all of them by studying how they are using RNA structure to do what they do. So it is true, though, that even though most of these do not infect humans, there are a lot of dangerous viruses that actually do. And many of these fall into a classification that we call RNA viruses. And what these means is that these viruses are so invested or dependent on the use of RNA that in many cases they have completely eliminated the use or the need for DNA anywhere in their infection strategy. They are RNA-based infectious agents. And in many cases, you can induce an infection just by taking the RNA out of the virus and injecting it into a cell. That is enough to create a viral infection. So I think it becomes pretty obvious that, in order to meet the pandemics of the past, the global outbreaks of the future, we need to know more about these viruses, and one important way of learning about them is to study their RNA, to study how they are using RNA to do different things, and then to use that information to develop new therapeutics. Okay, so that's a fairly, I think, important reason to study RNA and specifically viral RNAs. But there's another reason, another thing that motivates me that may be a little bit less obvious. And that's the fact that viruses on their own cannot make more copies of themselves. If I placed some virus on this podium and let it sit there, it would never multiply. It would never reproduce. It can't. In order for a virus to make more copies of itself, it must infect the cells of its host. The virus enters the cell and it makes use of the biological molecules within that cell, and it turns that cell into a little virus-making factory. So this has a really important implication. What it means, whoops, that was the wrong button. What it means is that it doesn't matter if you are an RNA that is made by the cell and you belong in the cell and you're on the RNA home team, or you're a viral RNA that the virus has made and has entered the cell and you're on the visiting team. The visiting team and the home team both have to play on the same playing field, and they both have to play by the same rules. And so what this means is by studying how viral RNAs behave and operate and do what they do within the cell, we learn fundamental principles of RNA biology. We don't just learn about the viral RNA, we learn how RNA in general works. And we can learn by studying viruses that infect humans, or ants, or sea slugs, or bacteria, or elephants, or any of the above. They all have something to teach us. And once we learn those fundamental rules, we can apply them to things like understanding cancer, understanding diabetes, or neurodevelopmental diseases. So I can say with great confidence that by studying the RNA from a virus that infects a cricket, we can learn things that could potentially be useful to develop new cancer therapies. Okay, so I'm passionate about RNA structure, and I'm passionate about studying viral RNAs. But how do we actually look at molecules? Because the way that I want to study these RNAs and that I want to understand them is how do their three-dimensional structures, how do those interact with other molecules, and how do they create all of these diverse functions? And so in order to do that, I need to look at molecules. I need to see their structure. So how do we look at something really small? This is audience participation time. High school biology, middle school, grade school, what did you use to look at something small? Microscope, exactly. We can, in fact, use microscopes to look at molecules, but probably not the microscopes that you have encountered in the past. So this microscope is not going to do it, not nearly powerful enough to look at molecules. You might turn to a microscope that looks like this, and there are many of these on campus. They're powerful enough to look inside cells, but they're not powerful enough to look at the three-dimensional structures of individual molecules. So we need to start using something called an electron microscope. But even one like this is not powerful enough. What you really need is a microscope that looks like this. And just for scale, you can see some of the researchers sitting down here next to it. That's how big this is. This is called a cryo-electron microscope. And as you can see, it's basically not really just an instrument. It's really a facility, right? And facilities like this are incredibly important. They are, and I'll point out, they really are beyond the resources of an individual investigator or even a couple investigators' labs to put together. This is the sort of facility where philanthropy and capital investment can really make a big difference in the research capabilities of a campus and really a region. So cryo-electron microscopy and these types of microscopes have been around for a while, but it's really in the last 5 to 10 years where they've undergone this amazing technological leap forward. And it's really only in the last 2 or 3 years where we've started applying this technology to RNA. And I think the potential is enormous that this technology may allow us to start routinely looking at the three-dimensional structures of RNAs, including viral RNAs, and making profound discoveries that would not have been possible before. And I will say with some pride, but also with some confidence, that I think there are 2 places in the world right now that are at the cutting edge of developing the methods to apply these sorts of technologies to RNA structure. And one of them is Stanford University, and the other one is the University of Colorado Anschutz Medical Campus. Yeah. All right, so I'll finish up here by just going full circle. So I showed you this structure near the beginning of my presentation. It turns out this is a piece of RNA from the Zika virus. And we started studying this RNA actually about a year before the global outbreak of Zika. We were motivated purely by curiosity. This RNA does something interesting, and we wanted to see the structure that gave rise to that interesting function. We had no idea it would become a global health threat. And so David said that basic scientists are thrill seekers. Indeed, we were thrilled to see this structure. But also, we were thrilled about the unexpected things that came out of this study where we never thought we were going to go, new directions that we never could have foretold. The structure revealed things to us that we are now partnering with another lab on campus, the Beckham Lab, to develop a vaccine platform against not just Zika virus, but all kinds of mosquito-borne viruses. We've engineered this RNA, and we now have a technology that's patented in the US and overseas, and we're hopeful that that technology will be the basis for all kinds of new therapies. And believe it or not, things that look kind of like this structure appear to be present in some marine bacteria that are important for the renewable energy field. And so we've been in conversations with people at the National Renewable Energy Lab about how our discoveries may aid their research. And so we're thrill seekers, but we're also explorers, right? And we set out on a path based on pure curiosity. And what we hope is as we move along that path, there are going to be unexpected discoveries and unanticipated benefits. And I think that's where the real power and promise of basic science lie. So thank you very much. Yeah. And so it is now my pleasure to introduce Jay Hesselberth, who is currently one of the co-directors of the RBI. And yeah, I'll let you, and Suja as well. Suja Yaganathan they'll be kind of going back and forth here, so.
Okay. Well, thanks everybody for coming. As Jeff said, my name's Jay Hesselberth, and tonight, Suja Jaganathan and I are going to tell you a little bit about RNA therapies that we've been developing on campus that have far more implications than the obvious ones. And I hope by the end I'll provide you a little bit more detail of how these RNA therapies work. So I'll start with a question that I used to get fairly frequently a few years ago. I'd get this question when I was on family vacations during the summer. I'd get the question from parents I'd meet at my kids' schools, or I'd get it from my barber. Whenever I would get this question, it was always fairly awkward for me. I'd quickly change the subject to housing prices in Denver, or my favorite musicians, or the weather. But in the meantime, RNA virus has turned the world upside down, and an RNA vaccine, in large part, saved it. So now it seems like everybody wants to talk about RNA at these events, and I'm happy to oblige. I'm an RNA biologist and computer scientist, and I have a dream job that can be described by this simple equation. So my job is to combine RNA sequencing data with blank to produce ideas that we can test in the lab. This is also audience participation. Does anybody have an idea what blank might be? I seek mine out at shops in the mornings. I do my best work there. African origin, a little bit of milk. Anybody? Coffee, exactly. So my job is so this is a timeless formula for success in science. Data plus coffee equals insight. So given this, it might not surprise you to learn that my work in RNA therapy started a few years ago when I had a chance encounter at the campus coffee shop. So I met a few colleagues from Children's Hospital Colorado who were excited about a new class of RNA therapies, but didn't know how to move the needle to bring those therapies to campus. This began a beautiful collaboration between basic scientists like myself and clinicians to hopefully impact the lives of children with neurological diseases. So what we didn't anticipate back then when we started the RNA Biosciences Initiative was how quickly RNA medicine would change the world. We've really only seen the tip of the iceberg. We've entered a new era of RNA medicine, and there's many opportunities in front of us. And we're here tonight to convey our excitement for those and try to give you a sense for where the RBI is headed over the next few years. So I'm going to tell you about our work in RNA therapies, but before I get into that, I'm going to let Suja introduce herself and tell you about her work as well.
I'm excited to tell you all a little bit about my background and the work my lab does at the RBI. Some of my earliest and most vivid memories are actually as a little girl, rainy evenings, sitting on my dad's lap, after dinner, listening to bedtime stories. Now, these were not your typical bedtime stories. They were stories of scientists and scientific discoveries. The context here is my dad's a scientist himself, a theoretical physicist, to be precise, and he told the most wonderful stories of scientific discoveries. So I learned from him about, a scientist who noticed that milkmaids who had previously gotten cowpox were immune to getting smallpox, and how vaccines came from that. Or how when somebody noticed that bacteria would not grow around a certain type of mold, and from that came the discovery of antibiotics. These stories really captivated me as a kid, and they were so simple. It's just somebody paying close attention to the natural world around them and asking simple questions: why not, and how? And when they were able to answer them, the knowledge they gained could transform the world. I was very taken with the idea of becoming a scientist, and really I don't think I've ever considered being anything else in my life. So I'm happy to report that some decades later I am a scientist. And I get to do this, I have a dream job where I get to ask questions, answer them, and do that for a living. And the topic that I have chosen to study has to do with a very basic process that happens in every living cell. It's a quality control process, actually. So let me introduce to you what I mean by that. We saw earlier a video about how the information that's in our cells is stored in our DNA, right? That's the genetic information. This is actually present inside of what's called the nucleus of a cell, where you can think of it as the command center of the cell. This information gets transcribed into what's called an RNA molecule, which then goes out into what's called the cytoplasm of the cell, which you can think of like a factory floor where proteins are actually produced. So the cell is much like a factory where a simple set of instructions are used to produce protein molecules that do the job by which life occurs, right? So if you think of this, it's basically instructions being copied to produce RNA, and then these copied instructions are delivered to the factory floor, and then protein molecules are made, right? Except that this process happens in an average cell in your body, you and me sitting right here. 2 million proteins are produced every minute, okay? So you can think of the scale at which this operation is going on in our body, and for a process like that to happen accurately, you need quality control. So if you have a good molecule that is a faithful copy of the information contained in the DNA, you do want to make the protein, and this process is basically, the quality control ensures that you make a faithful copy of the instructions in the DNA to make a faithful protein molecule. So if you think about how quality control works, you need people who, in a chocolate factory, this might look like people who make sure that everything happens properly and that the information, that the chocolate is wrapped properly and whatnot, right? But if you stop having good quality control, what ends up happening as you can see, is chaos, right? But in a cell, when quality control does not happen, it can be quite devastating, actually. You can actually have diseases that come because your cells cannot carry out quality control. I study one particular disease that's an example of this phenomenon called facioscapulohumeral muscular dystrophy. This disease, it affects muscles in the face, the upper arms, and shoulders. And people that have this disease become wheelchair-bound by the time they are, in their late adulthood. Another thing that happens is they lose control of their facial muscles, so they're often not able to express emotions or smile. This disease is actually quite prevalent. About 1 in 8,000 people across the world have it. And the key point that my lab is interested in is that in this disease, muscle cells in your body cannot carry out quality control. And this seems to cause the production of bad proteins in these cells. So knowing what we know about this disease, you can ask the question, how can we cure FSHD? And I want to take a step back and ask a slightly bigger question. How do you cure any disease, right? To answer that, I want to tell you a story. A few years ago, I was in the audience at a scientific talk where a scientist spoke about the work his lab had done for decades, trying to understand how little pieces of RNA were cut and pasted together in a process called splicing, and how the understanding that his lab developed over decades allowed them to produce a cure for a muscle disease called spinal muscular atrophy that allowed a small child to walk for the first time in his life. There was standing ovation. And let me tell you, that's not a thing in science. The reason I say this is because his lab was able to put in decades of work understanding this basic process that they were able to develop a cure for this devastating disease. This molecule is Spinraza, and the scientist is Adrian Krainer, and this is an RNA-based drug that is now hailed as a miracle cure for spinal muscular atrophy. So the point that I want you to take away from this is that it's really understanding a disease, understanding the basic biology of how cells work that will allow us to produce a cure. So what my lab is doing now is asking basic questions in FSHD, such as how does RNA quality control work? How does it fail in this disease? And when it fails, is there something we can do to prevent it from failing? Or if it's already failed, can we reverse the damage, right? These are the questions that we come into the lab every day and we ask. And if we are lucky, someday we'll get an answer that will allow us to actually develop a cure for this disease, FSHD. And with that, I'm going to turn it back over to Jay.
So as Suja mentioned, Spinraza, the drug used to treat spinal muscular atrophy, is a real miracle drug. It's improved the lives for over 10,000 people with spinal muscular atrophy. What my colleagues in the coffee shop, who I ran into, were particularly excited about is another RNA drug that was developed to help treat a little girl named Mila. And I was at the seminar that Suja was at, where Adrian Krainer got the standing ovation. My colleagues will also probably remember a seminar we were at where the story of Millicent was told. And while the standing ovation in Krainer's seminar was obviously quite joyous, the story of Millicent is even more profound, and that seminar is the only seminar I've actually cried at. So how can this be? How can you have two seminars where you're hearing the same story about RNA drugs but having two particular outcomes? So the story of Millicent is particularly remarkable for a couple of reasons. So the first is that the drug for, let me back up. So a couple of years ago, a little girl named Mila came to Children's Hospital Colorado and was diagnosed with a rare neurodegenerative disorder called Batten disease. So what's remarkable about the story of Millicent, first of all, was that the drug was developed with remarkable speed. So from the discovery of the mutation that caused Mila's disease to the delivery of the drug to Mila was about 10 months. So at the time, this timeline was completely unheard of. for the development of any drug. And it involved identification of the mutation, development of the drug in a lab that could correct the impact of the mutation, manufacturing, and even FDA approval. This last step can often take years. So the second thing that you should know about mealocin is that it's a drug that could only treat Mila's unique mutation. It's the first example of a truly personalized RNA therapy. So this is a picture on the right of Dr. Tim Yu, whose team at Boston Children's Hospital was responsible for spearheading the development of mealocin, as well as Mila and Julia Vitarello, Mila's mom. So tonight, Julia Vitarello and Scott Demarest are actually in the audience. Scott was Mila's physician here at Children's, and they've been truly spectacular collaborators on our efforts to develop RNA therapies. So Mila's condition, as well as a variety of human diseases, are caused by a process that Suja mentioned called RNA splicing, during which RNA molecules are cut and pasted together to create a template for protein production. So to describe this, I'll use an analogy to Legos. So the molecule you see here, the colored boxes, are going to be put together, and in the lab we call those colored boxes exons. And the intervening dashed lines in the lab we call introns. So what's particularly remarkable about this RNA molecule is that it not only contains the Lego blocks, but also the instructions to put those Lego blocks together. So when a cell sees this RNA, it's going to put the black, gray, and gold boxes together, and this yields a normal mRNA and a healthy cell. However, mutations that we sometimes inherit from our parents can impact this process. And what happens is those sometimes can cause the cell to misread the instructions, and instead of putting the black, gray, and gold boxes together, it puts the black, red, and gold boxes together, and this leads to disease. About 50% of human genetic disease is thought to be caused by problems in RNA splicing. So if you've ever spent any time with children, you've probably had the experience of dumping 1,000 Legos onto the floor and reading the encyclopedic instructions for putting a Lego assembly together brick by brick. This takes hours. I've done this a lot with my kids. But what can often happen is, let's say on page 47, you stop paying attention and you forget to include a couple of key pieces into the assembly. So this is the difference between producing a sleek Millennium Falcon on the top or a Millennium Falcon with a small problem. It's missing the chairs for Han and Chewie to sit in. So now you've got a broken Lego and an extremely frustrated 5-year-old. This kind of gives you a sense of what the cell experiences when it sort of misreads the instructions to put the mRNA together. So it turns out what's really interesting about these RNA therapies is that they're pretty easy to design. So if we go back to this example, this mutation, if we zoom in on that particular mutation, you can see that it's just a single position in a string of RNA letters that's changed, that red C. So the RNA drugs in blue here, have been developed. Their role is to seek out those mutations in cells, find their targets, and essentially mask the mutation from the cell. So in doing so, they prevent the cell from misreading the instructions, and they promote normal splicing. So while these drugs can target RNA sequences, I think it's pretty easy for you to see how you might also be able to design RNA drugs that target RNA structures, like the ones Jeff mentioned, that are so important sometimes for RNA function. So it's really the flexibility of this platform that has people so excited. So to bring these therapies to campus, we started a collaboration with Scott Demarest and his team at Children's Hospital to develop RNA therapies for kids with rare neurogenetic diseases. In the pipeline we developed starts with identifying a patient that might have a mutation that could be targetable by RNA therapies. Once we find those, we generate a renewable resource of cells that we can grow in the lab from a blood draw. and then characterize those cells via RNA sequencing to look at the impacts of that mutation on splicing, and we can use all of those data to design drugs and try to correct the impacts of the mutation on splicing. So to date, over the past year and a half or so, we've applied this pipeline to about a dozen kids with diseases ranging from epilepsy to neurodegenerative disorders. And we've discovered a lot of changes in splicing that are caused by their particular mutations. We've also found that this process is incredibly difficult, and we've met with a lot of failure, but we still continue on. And one good thing that came out of this is it forced us to start thinking about other ways to apply RNA therapies to help these kids. And so to touch on this last point, I just wanted to bring up one of the final things that is sort of the elephant in the room when you think about personalized medicine, which is that while developing RNA therapies is certainly exciting, the reality is that developing an RNA drug that's specific to each individual mutation is quite a daunting challenge and quite expensive. So each of these today costs about $1 million each. So in the US, there's about 25 million people that are affected by a rare disease of some kind. And many of those have a genetic component that might be targetable by a melisin-like approach. So you can see how that process applied to hundreds or thousands of people with these rare diseases is sort of it's really not a very scalable problem. So we started thinking about how can we make fewer RNA drugs that help more people. So our work to date has focused on the development of what we're calling socialized or democratized RNA therapies. And this is one of those thrills that David mentioned early on. One of my favorite things as a scientist is to take an idea on paper to an experiment that tells you something might actually work. So these therapies we think might be able to address a problem in genetic disease called haploinsufficiency, which is just a fancy way to say that normally people inherit 2 copies of a gene, one from mom, one from dad. But in the cases of haploinsufficiency, you only inherit 1 normal copy. The other one's broken in some way. And that 1 copy is not enough for normal cellular function. So in this example, I'm just showing you 3 people that have one unique mutation in each copy of the gene, which necessitates the development of 3 different RNA drugs. Okay? So now you can imagine the case where you might not have 3, but 30, 300, 3,000 different mutations. Again, this is not a very scalable problem. So what we realized is that in each of these cases, all of these people have a normal copy of a gene. And what we've been able to do is develop RNA therapies to tell cells to just make a little bit more of each of these normal copies, okay? And not worry about the individual mutations, just focus on the gene itself. So we've been calling these silver bullet RNA drugs in the lab because the development of 1 drug can help 3, 30, 3,000 people potentially. And in doing so, we greatly simplify the drug development process and hopefully provide a new approach to these genetic diseases. So I hope that I've given you a little bit of insight into how we're developing RNA therapies here at CU Anschutz. We've only given you a glimpse of the RNA therapies that are currently being developed. There are many others, including RNA vaccines, as well as RNA therapies for other diseases such as cancer. Suja and I have both told our stories, and we hope that you see how the development of RNA therapies at CU Anschutz is progressing along nicely. So it's my pleasure to introduce the final speaker of the evening. Olivia Risland is going to tell you about the development of messenger RNA therapies.
Great. Well, thank you for that introduction, Jay, and thank you all for coming this evening. What I'm going to be talking to you about is a different type of RNA therapeutics called messenger RNA therapeutics. And probably of all of the RNA therapeutics out there, this is probably the one that you have heard about before coming into this room. Because messenger RNA therapeutics, or mRNA therapeutics, form the basis of the Moderna and Pfizer COVID vaccines. But what I'm hoping is that you will get 2 key points from my presentation this evening. The first is that the potential of mRNA therapeutics extends well beyond COVID-19 vaccines. And point number 2 is that actually we need to understand more about the fundamental biology of RNA to realize that potential. Now, where I want to start is actually not with RNA, it is with Star Trek. Because when I was a kid growing up, my father and I used to watch Star Trek, The Next Generation, of course, every weekday evening at 7 p.m. And perhaps it's not so surprising, given the career that I ended up choosing, that one of my favorite characters was Dr. Crusher. Now, Dr. Crusher was the chief medical officer on the Enterprise. She was also the chief biomedical researcher. And some of my favorite episodes also involved her really being the star of the show. And these episodes would often unfold something like this. The Enterprise would encounter some new pathogen. It would affect a member of the crew. It would spread throughout the Enterprise, and one by one, the crew would start, succumbing to this illness. And it would be Dr. Crusher's job to save the Enterprise. So how would she do this? Well, step number 1 was figure out what the pathogen was. Was it a bacteria? Was it a virus? What was that? Step number 2 was figure out its vulnerabilities. How are you going to kill the pathogen? Step number 3, create a cure. Step number 4, figure out if this potential cure would work. And step number 5 would be to administer that cure to the whole entire Enterprise. And she would do so over the course of 1 episode of Star Trek. Amazing. And in Star Trek time, this is like 1 or 2 days. And to be honest, right, this seems like fiction, science fiction, in fact, right? That you could go from having your first interaction with a pathogen and then having a cure a day or 2 later. And I think what we are all realizing in the RNA community is that maybe this is not such science fiction. And the reason that I say that is because some of how quickly some of these RNA drugs have been developed. So if we think about the example that Jay brought up of Millicin, it was 10 months from identifying the mutation to treating Mila. If we think about the COVID-19 vaccine, this is actually even more amazing. So the first genomic sequence for the virus came out in January of 2020. By March of 2020, so just 2 months later, Moderna was injecting humans as part of its clinical trials. 2 months. That's amazing. And part of what makes this possible is that RNA is easily programmable. It's actually very easy to make. We know how to make a lot of RNA because, again, of decades of research. And so all we need to do is just figure out what the RNA drug is. And so I think about this a lot like building highways. We're pretty good at building highways. We know how to build highways. The real art to building a highway is not the building of it, it's the design. We have to figure out where is the highway going to go, how many lanes is the highway going to have, where are the on-ramps and off-ramps going to be. The art of mRNA therapeutics is not the production, it's in the design. So the big question that RNA researchers are really wrestling with is, well, how do you design an mRNA therapeutic? And it turns out that to answer that question, we actually need to answer 2 slightly smaller questions. We have to understand what an mRNA does, and we have to understand how it does that. So what does an mRNA do? Well, to get there, we actually have to take a step back and think a little bit about what is actually in our genome. So I think of our DNA and our genome a lot like The Joy of Cooking, the cookbook. Joy of Cooking in your kitchen probably has all of these recipes to make all of these different meals. It can make a delicious Thanksgiving meal. It can also make my favorite breakfast of pancakes and bacon. Has all of these different recipes. And when you're cooking, you're choosing what recipe you're going to use, and that choice will determine what meal you're going to put on the table. The exact same thing is true in our cells. What makes a skin cell different from a kidney cell, different from a liver cell, is not the cookbook, it's the recipes that are used. Now, of course, the DNA cookbook is a lot more complicated than The Joy of Cooking. So our DNA has about 20,000 different recipes, and each cell in our body is making and following thousands and thousands of recipes all simultaneously. And again, if we just take a moment to think about the complexity of this, each of us in this room has about 14 trillion different cells. Each of those cells is following thousands and thousands of recipes. This is a very complex, complicated process. But there's one really core idea that is the basis of mRNA therapeutics. And that is the idea that an mRNA is essentially a cellular recipe. Why is this idea so powerful? Well, if we come back again to thinking about genetic diseases, like the case in Mila or say in the case of cystic fibrosis, there is a problem in the DNA that makes that recipe not work. So the cell can't produce a dish that it needs to make. An mRNA is a recipe. It doesn't matter where that mRNA comes from. So if we can just give the cell the recipe, it can make the dish that it needs to make. So all we need to do is just make that RNA. Now, if you actually ever talk to any of my students, they will tell you that the minute I say something like, well, all you need to do is blah, 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 that is immediately a clue that this is going to be a really hard problem, that it's probably going to take much longer than any of my estimates. And it's true, this is a really hard problem. And this is a really hard problem because, again, let's go back to thinking about meals that we cook. If you think about Thanksgiving, right, it's not just what dishes you make, it's when you make them, right? So you want the turkey to be done before the apple pie. You want your cranberry sauce to come out with your turkey. It also really matters how much of the dishes you make, right? You want to make sure that you have the right amount of Brussels sprouts. In my family, that is a lot of Brussels sprouts. So you need to make the dishes in the right place, at the right time, in the right amounts. And the same is true in the cell. Cells need to follow recipes in the right amount, in the right place, in the right time. And the real power of mRNA is that in addition to having this recipe, it also has all of those additional instructions about how much, when, and where. What does an mRNA recipe look like? Well, this is a cookbook recipe for apple pie, right? So what is the corresponding mRNA recipe? Well, now I want to remind you something that you've heard about a couple times this evening, which is that RNA is made up of a string of nucleotides, a string of letters, A, C, G, and U. So our English alphabet has 26 letters. RNA alphabet has 4 letters. But just like English, it is the order of the letters, that linear sequence, that is what conveys the information. So while a cookbook recipe might like this is what an mRNA recipe looks like. And so obviously, if we're going to start designing mRNAs, we need to be able to understand what this syntax is. We need to be able to create a Rosetta Stone, right, so that we can understand what this language is. Now, if we think about a cookbook recipe, there are different components to it. We have this bit here, which tells us ingredients that are going to be combined into the apple pie. We have this section here that's going to say, okay, this is where the recipe starts. This is where you start putting the ingredients together. And then this bit here is going to tell us how we're going to combine the ingredients. What order, how much, how long, all of those things. The exact same types of information are contained within mRNA. So in this example, it is this sequence here that is telling the cell what ingredients are going to combine together. These 3 letters, AUG, tell the cell this is where to start. And in fact, in mRNA, AUG is a very, very special sequence of 3 letters. This tells the cell this is where the recipe starts, all the time. And to be a little bit more precise, it is this AUG that tells the ribosome, the machine that makes proteins, okay, here is where you're going to start making the protein. This sequence here tells the cell to not make this dish when there aren't a lot of nutrients around. We know all of this about mRNA, again, because of decades of fundamental research in RNA biology. But there's actually a lot that we don't understand. So, for instance, we know that there are other bits of information in this RNA that are telling the cell when it should make this dish or when it shouldn't make this dish. So we know that there is information here. We don't really know what that information is. We think that it might have to do with structure, like what Jeff told you about, but we don't really know. And something that my lab works on is actually more information that is contained down here, where again, there is information that's telling the cell when, where, and how much of this dish should be made. But again, we don't really quite understand this yet. And to make really effective mRNA therapeutics, we need to understand the syntax of these cellular recipes. So what I hope I have convinced you of tonight is that mRNA therapeutics have this huge potential where they could cure many different genetic diseases. To realize that, we need to understand the syntax of these cellular recipes. And there is still a lot that we don't understand, and this is what many of us spend our days and nights thinking about. And we do so with the motivation that if we can understand these fundamental principles of RNA biology, that someday this could really make a difference for human health. But one point I want to end on is that it is not just that motivates us. As I think you've heard throughout many of the talks this evening, a huge motivation for us is actually discovery and exploration and understanding the mysteries of life. So I remember the first discovery that I made, like the first real scientific discovery that I made. I was in graduate school in Oxford in the UK, and it was the summer of my first year. And a couple weeks earlier, I had had this sort of wild idea of an experiment to do with my favorite protein. This was the protein that I had spent, the whole last year thinking about. And I was going to do this wild experiment. But before I could do the wild experiment, I needed to get my ducks in a row to make sure that even technically, this thing had legs on it. So it was a Friday afternoon, and all of my friends were at the pub, because of course they were, because we were in England. And I'm in a pretty dark room that was very heavily air conditioned, seeing the results of what I thought was a very banal experiment. And the way that this particular result appeared was I had a gel that was being scanned, and it was coming on the computer screen sort of like pixel by pixel, like those old school printers that used to come out. And so I was seeing it slowly being revealed before my eyes. And I remember this instant where the result came out, and I realized that everything I thought I knew about this protein was wrong. Everything that I had been thinking about for the past year was incorrect. But there was more than just that. It was like, not just what were we incorrect about this protein, but we were actually missing a whole puzzle piece of RNA biology. This whole entire vista about how RNA is destroyed in cells, we had just missed. And I was the first person in the history of humankind to know that fact. It was awesome. And I think for a lot of us, it is that potential to discover, to understand a mystery of life, to have that moment where you really get to see something new. That is a huge motivating factor for us. And it is really, to be honest, one of the great joys that I have in my job. So, okay, Star Trek, yes, it is fiction. We are never going to have warp drive. We are not going to have transporters. But I would suggest that the things that Dr. Crusher would do, the speed with which she would come up with cures, I would suggest that all of that is a lot closer to reality than I thought as a young girl. So with that, thank you, and I think we'll be taking questions.
Our speakers have made the assumption that the 4 nucleotides that are responsible for RNA, G, C, A, and U, are similar, but I want to point out to this audience in particular that there's a hierarchy, and 2 of those nucleotides are more important than the other 2. And so I ask this audience to, for the moment, suspend reality and think not about G, not about A, but think about C, U. Again, as promised, we have time for some questions, but I would like to introduce other members of the RNA Bioscience Initiative who are here with us this evening who didn't speak. So feel free to stand or not as I call out your names. Aaron Johnson, Neil Mukherjee, Matt Taliaferro, and Srinivas Ramachandran. I have no doubt that if you go up to any of these people after this evening is finished and ask them what they do, that they will just talk your ears off. So let me invite our speakers back on the stage, and I'm going to give you the opportunity to ask them some questions. So while the audience is contemplating questions, let me take the prerogative of the chair and ask the first question. And I think, as a couple of the speakers have said, it's fashionable to say that RNA saved the world. Thank you very much. Of course, I'm speaking about vaccine development, but I wonder if some of you could just summarize why all of a sudden the field of vaccines changed so abruptly. We've been giving vaccines to things for years and years and years, and suddenly everything switched. And RNA vaccines became truly innovative. That's the first part of the question, the why RNA vaccines. And the second part of the question is, what's next? So we've intimated vaccines for a variety of infectious diseases, but my question, are we going to have vaccines for Parkinson's? Are we going to have vaccines for cancer? For other diseases that are going to affect large numbers of people in this audience? It's a two-part question. Go away.
So I'll start with why mRNA vaccines are so important. So if you think about the history of how vaccinations worked, there's a couple of different approaches we've used over 100 years plus to vaccinate ourselves against infectious disease. So one is live virus, or attenuated virus. Those are sort of hard to grow in the lab and can be quite dangerous. Things like subunit vaccines, so components of viruses, proteins, we put into people. You might know that, for example, the influenza virus is grown in chicken eggs. Also, obviously effective, but not very scalable. What's amazing about the mRNA vaccine is it's solved a couple of different issues at once. So you can make the mRNA in large quantities with no problems at all. You don't need chicken eggs. You just need a big, giant molecular biology reaction to make a lot of mRNA. And the second is the mRNA itself not only encodes for the antigen, the protein that our bodies are responding to, but also is the basis for getting our bodies to respond. So in and of itself, creates sort of an inflammatory response, leading to the sort of the potent efficacy of the mRNA vaccines. I actually missed the second question. Like, what are other applications? Is that?
Yeah, you're going to cure cancer?
Yeah. So the problem with cancer, of course, it'd be great if we had a vaccine for cancer. One of the problems with cancer is that we don't know what the target is to be made often. So while the mRNA vaccine encodes a portion of the spike protein on coronavirus, in the cancer case, we often don't know what the body needs to make antibodies against. And so that's really a big challenge right now. I would say the same for Parkinson's, or basically, lots of other diseases.
Since you brought up cancer, I can actually make one point, which is that there's a really interesting way of fighting cancer recently that takes advantage of the stuff I work on, quality control. If you can actually make cells be bad at quality control, the body will attack those cells because they start making proteins that have no business being made in the cell. So people are actually thinking of cancer therapies where you specifically inhibit quality control in cancer cells, and by doing so, you make them immunogenic. And the body attacks them and kills them, right? This works in mice. Of course, we have a long way to go to make it to work in humans, but I just want to bring that up as, things that you may not expect to have an application. You'd be surprised how far some of these things go.
Thanks. Do we have questions from the audience?
Peter, I was just going to say
Yes.
one thing regarding the first part of your question about the timing of the development of the RNA vaccines against the COVID virus. The idea for RNA vaccines started out in the 1990s. It was a totally wacky idea, a crazy idea that very dedicated, creative people stuck with and proved was actually correct in the face of a lot of skepticism. I believe it was in some ways a happy coincidence that after 30 years of basic research developing this crazy idea, by 2020, that technology was mature and ready to put into action just when the world was challenged with the deadly threat of SARS-CoV-2.
This is not a strategic level question, but I was intrigued when Dr. Keese was talking about electron microscopy. How do you know when you're trying to find out the structure and order of things in an RNA molecule, how do you know that you're looking at the one that you're actually interested in?
So that's a good question. So the way that most electron microscopy is done now, the majority of it, is that we actually go through a pretty stringent purification process to get the molecules that we want. And then we have to process them in a certain way to get them on a really tiny grid, and we freeze them in a certain way, and they go into the microscope and so on. And so it turns out to be a very good structural biologist, you need to be a very good biochemist, right? It isn't just about having a microscope, it's about knowing how to work with the molecules and make the molecules and purify the molecules. However, I'll say that more and more we are seeing the ability to study the structure of molecules inside cells. So to actually take cells, put them into the microscope, and then use various techniques I won't get into in detail to identify the molecules you want to look at and hone in on those with the electron microscope. That's extremely exciting. We call that tomography, and there's various other applications there. But it means you don't have to purify, you don't have to pull it out of the cell. It's where it would normally be in the cell in kind of a state of suspended animation, for lack of a better term. And you're looking at what it's doing, what it's interacting with, and its state in the cell. That's really the cutting edge right now. But these sorts of microscopes are allowing those sorts of experiments. So it's really exciting.
Yep. So in the world of research funding, is it more difficult to get funding for basic research than it is for applied translational research? And if so, why would that be?
Yes.
And then why would that be?
Yes. So I think one thing to understand about how biomedical funding tends to work in the U.S. is that the major funder is the National Institutes of Health. This is a federal government, essentially it's run by the federal government. So taxpayer money funds research, both basic research and translational research. And I think this highlights maybe a challenge that the NIH has, which is what should their portfolio look like? How much of their budget should go to designing things that could help patients next year versus basic research where that timeline might be 30 years? And I think this is really the challenge that funding agencies have, which is how do you balance that? And it can be really tempting, especially when you see people who are suffering from devastating diseases, to want to help those people. I mean, that is a human emotion, a human impulse. And it is actually hard to remember that basic research essentially starts the pipeline for the cures that are going to come in 20 or 30 years. And I think it is that tension that can sometimes make it a little bit challenging to get as much funding for basic research as perhaps we might like.
One of the things that I'll augment what Olivia said is that timeline actually has shrunk. And that's one of the most astounding things about the last decade of RNA biology, which is that the pipeline from discovery to application, that time is shortened, which is just a remarkably exciting development. Do we have other questions? Oh, for heaven's sakes. Come on, audience. Okay, let's. I want to thank our extraordinarily enthusiastic and remarkable speakers. I want to thank the Office of the Chancellor for putting on this program and the School of Medicine Dean's Office for supporting the RNA Bioscience Initiative. I'll take a little bit of credit for that. And I want to thank all of you for coming to this important and really thrilling evening. Thank you very much.